This is the pre-lab for the plant physiology lab, which this semester will be entirely online. How do plants move water from their roots to their leaves? So, several goals for this week, including understanding how we connect Bio-1 type material, diffusion, adhesion, cohesion, to plant physiology. We're going to analyze some data, and we're going to identify the structures involved in the transport of water. So, plants, just like animals, have lots of chemical reactions, lots of functions going on in their body. The study of physiology is the study of function. You began this study when you learned about photosynthesis in Bio-1. Plants also undergo tropisms, which is a type of physiology. This is when plants grow towards the light or their roots grow downward, how they sense the world around them, how plants communicate with each other, how their cells communicate with each other. All of these things fall into the realm of plant physiology. We are going to focus this week on the movement, the transport of water, and a little bit about the transport of sugars as well. Several activities are planned for at least when this lab is in person, but I'm going to talk to you about what you would have seen. We would examine root hairs, stems. We would do a paper maceration. That's when we soak toilet paper usually, or paper towel, in a little bit of water, and we tear it up and we look at it under a microscope to see what it's made of. We would do a leaf peel. That's when you put clear fingernail polish on a leaf and then make a slide out of it to see the surface of the leaf. Prepared slide of a leaf, and we would run an experiment on transpiration. So let's start with the plant organs. At one end of the plant, you typically have leaves. Leaves are usually large, broad structures that are meant for the capture of light energy, and they have several tissues that are kind of important. So let's talk about some of those tissues. So first of all, we have an upper epidermis and a lower epidermis, as you can see in this drawing here. So there's an upper and lower epidermis, and in that epidermis are these special structures called stoma is singular or stomata is plural. These are structures that are composed of two guard cells, like we see here in yellow, and a stomatal pore, which is just an opening. And those guard cells can fill full of water and swell up and open, and that opens up the stomatal pore, allowing for gas exchange and for water to evaporate out. And then typically at night or during a drought, those pores will close. The guard cells lose their water, and the stomatal pore will close. So stomata can open or they can close, and they're involved in gas exchange. So we can see that here in the lower epidermis of this leaf. Now, the leaves also have a tissue called mesophyll, which you can see written here, mesophyll. There are two layers of mesophyll. The palisade mesophyll. Palisade means column-like, so they look kind of like columns. They're long and skinny. That's where most of the chlorophyll is for the light reactions. Down here is the spongy mesophyll. These cells have some chloroplasts as well, but there are fewer cells down here, and they're kind of round and they're spaced out. There's lots of air pockets, lots of surface area for gas exchange. So this tissue here, its primary function is gas exchange with the environment, kind of like the lungs of the plant. Also, within this mesophyll tissue is another structure here called the vein, and veins consist of xylem and phloem tissue that transport water and sugars for the plant. You've all seen the veins in a leaf, right? That's what they are. They are the xylem and phloem of the plant. Okay, so that's the leaf. If we were to look at a real leaf picture in cross-section, we would see the spongy mesophyll. See the air spaces here? Here's different stomata. You can see the openings there into the leaf. And up here is the palisade mesophyll, these long, skinny cells up here. And right there is a vein. And remember, this is in cross-section. So we cut the leaf in half with a razor blade, and we're looking at it under a microscope here. Now, at the other end of the plant, we have roots. Here's a root cut in cross-section. It has xylem and phloem. Xylem, these thick cells here, act like straws. They run the length of the plant. There are lots of cells that end up end to end together, like a bunch of drinking straws lined up from the roots to the leaves. That tissue transports water. Phloem is this thinner walled tissue out here. Phloem transports the sugars of photosynthesis. Now, on the outside of the root, you can see these hairs. These are called root hairs. They're single cell projections of the epidermis that greatly increase the surface area of the root, allowing it to absorb lots of water. Plants will also form symbiotic relationships with fungus, forming what you may have learned about in lecture, mycorrhizal associations, when fungus also grows into the root, increasing its surface area, allowing them to absorb more water. So the root's primary functions are to hold the plant in place and to absorb lots of water. Now the stem is the structure between the leaves and the root, and we're just going to focus on this tissue, these structures here. This is an herbaceous stem, so like a weed stem or something like that, something that's not made of wood. And these red cells are the xylem. Once again, we're looking at this in cross section. These cells aren't all that long, but they're cells that line up end to end, like I said, like drinking straws stacked up all the way up the length of the plant. That xylem transports water. And then this thin tissue here, that's the phloem that would transport the sugars. Now, over here is a woody stem, like a tree. And the rings of a tree, all these cells here, that's all xylem. That's water conducting cells. And they're really thick cells, so they give the tree support and strength. And the phloem of the tree is this blue tissue out here towards the bark. So woody stems develop a different anatomy than the herbaceous stems. So if we zoom in on xylem, it usually stains red. The lignin and cellulose stain red when you stain these things. So here's what it looks like unstained. You can see that the xylem cells often have these weird red rings. What this is thickened areas of cellulose and lignin that give those cells thicker cell walls so that they are supportive and they're strong. And so what we see here are several xylem cells lined up end to end, and water would be transported up these cells. And here's what they look like when you don't stain them. You can see that kind of corkscrew shape of those extra thick parts of the cell walls. And once again, lots of cellulose there. We learned about cellulose in Bio 1. That's what plant cell walls are made out of. But they also have lignin, which is like when you add rebar to concrete. It's a extra strength addition to the cell walls to make them extra strong. So that's your xylem. Now, what we would do in class is we would either go get some tomatoes from the greenhouse, or we would get some privet from outside. We'd cut those plants and we would bring them back into the lab. And then we would trim them and we would set up a potometer, which I'll introduce you to in a moment. So potometers look like this. So you take your plant and you stick them into this tube, and this whole apparatus is filled with water, and you have a syringe so you can add more water if you need to. So there's no air bubbles in this structure. This plant is down in there, and then right here is a scale in centimeters, and you fill it up until the water level is right at this 0 mark, and then you watch it over several minutes, and if the plant is using water, water will evaporate out of the leaves, and this water level will fall over time, and you can measure how fast water is lost from the plant. So why would water be lost from the plant? Well, this is the process known as transpiration. Your book may talk about like cohesion, tension, and these different words that all relate to this topic. So they talk about like cohesion tension theory, for example. So here's how this process seems to work. Water comes into the roots, which we don't have in this plant. Obviously, we trimmed the branch off, but water comes up the stem in this case, and it forms a column all the way up the xylem, all the way up to the leaves. Water molecules are evaporating out of the stomata if they're open from the leaf, because that's what happens. There's lots of water in the leaf is using water, and some of that water also evaporates out. Water is adhesive and cohesive. So it's adhesive, so water is sticking to the polar cell walls of the xylem. That helps the water column to keep from collapsing. But it's also cohesive. Water sticks to itself. So anytime a water molecule evaporates out of this leaf, it pulls at the whole water column behind it, and it pulls the water column up as those water molecules evaporate. And that's what pulls water up the plant from the roots to the leaves. That's what transpiration is. That's what cohesion tension is. Water is cohesive, and as it drifts away, it provides tension to the water column. It pulls the water column up as water evaporates. So if you were to set this up in lab, you'd have to be careful to make sure you had everything sealed tightly and everything was secure. So what you would have to do then is to write a hypothesis. So a research hypothesis, which would be if and then, that's your biological hypothesis, what you think would happen. And I'm having my students typically leave out the and part, so just the if then. So if is your explanation as to why something would happen, and then after the word then is what you think will happen in your experiment, your prediction. So if followed by the word then. But you can also do it in the traditional if and then format. So after if is your prediction, after if is your hypothesis, which is your explanation. After then is your prediction, what you think will happen if your hypothesis is correct. So then, once you have that written, you can work on your statistical hypothesis. And I'm just going to require a null and an alternative. So the null hypothesis, what would that be? What would H1 be? So what is the research question? Before you write your hypothesis, you have to know what question you're addressing. Well, the question is, do plants lose more or less water to transpiration if they're in a humid environment? So if there's lots of water in the air, if it's very humid, will that speed up or slow down transpiration? So you can work out your null hypothesis and your H1 there, as well as your biological hypothesis. What do you think will happen? And that'll be something you put down in the worksheet. So the way this would work is you would test this plant without any humidity around it, just room air, and then you would do it again, and you'd put a plastic bag over it, and you'd spray a bottle of water in there every minute or two, and you'd record how much water was lost over 5 minutes. Then at the end of the experiment, you'd dry off the leaves, you'd pick the leaves off, and you'd get their mass. And every table would divide their data by the mass of the leaves. That way we can standardize data. Because if one table has 20 leaves and another table only had 4, they would get very different rates because there'd be lots of different numbers of stomata between those two tables. So we have to standardize it somehow. So you would run it for 5 minutes, and then you would divide everything by the mass of your leaves, okay? And then you'd clean up and you'd be done. So that's what the data is on the worksheet. It's how many centimeters of water were lost in the humidity versus the control experiment, and that number is divided by grams of leaf, okay? Now, I want to point out that you'll need to run a paired t-test on this one because the same plants were used for the high and low humidity experiment, the same plants. So this is a paired t-test. So keep that in mind when you're completing your test. Here is what the leaf peels would look like. I told you we'd make a leaf peel in class where we paint leaves with fingernail polish so we can see the stomata quite clearly here. This is a student picture from 2 semesters ago or so. You can see those stomata clearly on the surface of that leaf. Also, this is what toilet paper looks like after you've macerated it. We can see these big cells here. This big cell here is called a vessel element. That's what flowering plants have in their xylem to transport lots of water at once. And these skinnier cells are tracheids. These are also water transport cells. They're found in flowering plants, but they're also found in conifers and other gymnosperms. In fact, conifers don't have these bigger vessel elements. Okay? And here's another example of a vessel element. These are, once again, great big wide cells that line up end to end in a flowering plant to transport water. And we can easily see that that's what paper is made out of, are these fibers and other parts of xylem. So xylem actually has several cell types, vessel elements, tracheids, and fibers that give it its strength. And you can still see them by just taking some toilet paper and squishing it under a microscope. It's pretty amazing. There's just another image of a plant cell with some spiral xylem that a student took a semester or 2 ago. Okay, so your assignment is simply to write your hypotheses, so a biological hypothesis, and then your statistical hypotheses. Then make a figure. That's a caption and graph. Run a t-test, a paired t-test, and then interpret what happened and why. So a short conclusion. That is your assignment, and I don't think it should pose too much of a problem for you. If you have any questions, email your lab instructor.